I'm Richard Csaky. I'm doing a PhD at the intersection of neuroscience and machine learning at the University of Oxford. I recently started the Oxford Science podcast to learn more about the diversity of science done in Oxford and the people behind it. The aim is to have long-form, open-ended conversations and delve deep into the science. Today, I'm very honored to have Mark Woolrich as the second guest on the podcast. Mark is a professor of computational neuroscience with a focus on developing new machine learning methods for neuroimaging data, such as fMRI, MEG, and EEG, to understand both the function and dysfunction of the brain. On top of doing research, he helped develop FSL, a software library for MRI data analysis, and more recently OSL, a similar library for MEG data analysis. He is also the director of the Oxford Centre for Human Brain Activity, which is part of the Wellcome Centre for Integrative Neuroimaging. I would like to thank you very much for agreeing to have this conversation, and also feel free to expand on anything that I omitted from your introduction.
No, I mean that was very comprehensive, and thanks for inviting me to have a go at this podcast. I'm intrigued to see how it pans out.
All right, when we can get into the neuroscience of it. So I'd like to ask broad questions. And one of the first questions that came to my mind when I was preparing for this is, what do you think is the goal of neuroscience? If you could...
Yeah, you weren't lying about going broad. The obvious answer, I guess, is it's the understanding the way in which the brain's structure and its kind of underlying biophysics leads to cognition, whether it's perception, movement, emotion, decision making, learning, etc. It's about understanding that relationship between the actual structure of the brain and how those cognitive behaviors pan out. So that's kind of the broadest definition that I would.
So it's a kind of
say
mapping between these, or is it more than a mapping?
It's providing the kind of explanation of how these cognitive functions arise from the actual physical entity that is the brain.
And what does it mean to understand? To really understand what's going on?
So, well, that's a deep philosophical question, I guess, which applies to any aspects of science and searching for knowledge. Do you mean that broad level?
I guess for the brains specifically.
As a kind of like the holy grail is to be able to have a representation that is able to generate the same behavior of the brain, where you understand what that representation is doing. I don't think that's really possible with the brain, but I guess that's kind of normally the objective in science. So you're looking at physics, for example, you try and come up with essentially a hypothesis or a combination of hypotheses that form normally a mathematical model that then makes predictions about the system. You see if your reality matches your predictions. And so the understanding there is in the form of the mathematical model, which is a representation of some rules that you've come up with, hypothesized, and then tested. So ideally you do the same thing with the brain. I just don't think that is possible to the same extent.
How much can this be applied to the brain? This kind of framework?
Not very easily. This is kind of why I've ended up working quite a lot at the level of sort of more somewhere slightly more abstract, I guess, in terms of representations of brain activity, rather than there's a bunch of people that work on trying to really push the boundaries of the biophysical models of the brain, so the physical modelling of the kind of connectivity in the brain and the different neuronal populations and how they interact down to the level of firing rates and conductivity of synapses and this kind of thing. And that's an impressive thing to try and do, but I'm not convinced that it's going to get you sufficiently near to the truth to be worth doing, at least using current tech and current compute. So I kind of sort of pull back from that slightly and work on models which are a bit more descriptive in their nature. I mean, I think I've heard some people describe things like the brain as like computationally irreducible. So if you really want to have a physics-like model of the brain, it kind of is the brain.
It's the brain. It's the smallest thing that.
So you end up, you might come up with a fancy, mathematical version of that you can implement in silicon, but it's not going to be anything really different to the thing you're going to be studying. So you won't get any insight from that.
But what you're doing is also, you said descriptive, but also predictive, right? Yeah. Would you say that's what understanding is? If you can predict certain behaviors?
I mean, it's a level of understanding, of course, but it's not. I don't think it's the kind of understanding that people sort of really think they're seeking in science typically. It's obviously a more pragmatic approach, I would say. It's like, and a lot of science actually is about being pragmatic rather than finding the kind of getting nearer and nearer to this unreachable truth that's somewhere out there that we never get to. So it's more about being pragmatic. Ultimately, I think all science ultimately is just pragmatic. Just as the mechanism of science is a good way of finding things that can be useful.
Yeah.
Because it kind of anchors itself on finding things that are reproducible. And if they're reproducible, they can be used reliably to achieve some pragmatic goal. But I mean, I'm not an expert on physics, but you could argue even the models in physics, they're not. You could argue even that's not computationally reducible. It's also a computationally irreducible problem, really, at the end of the day. Just that you find, I think in physics there's more, God knows why, the nature of the universe is the way that it is, but there's more like little problems you can work on where the problem is computationally reducible in a way that's useful.
To come up with a simple equation.
Yeah, whereas with the brain, I don't think that, so far, that hasn't really panned out quite so much, apart from at the very crude level. I mean, people do, I talked about the neuronal modelling that people do.
Yeah, you can reduce a neuron, right?
People do computational reducible versions of the reality, and they know they're doing it, and it's like mean field models of neuronal populations interacting. And that's is able to predict the sort of activity in the brain to some extent that actually is happening, but it's pretty crude and not as good, I would say, as the kind of non-biophysical models could achieve. And it's not clear. There's not really clear evidence that they're that much more useful from this pragmatic point of view I was talking about a few minutes ago.
Is the issue there the model itself or the scaling up of these models to the level of the brain?
I think the issue is just the sheer complexity of the brain and the level of. I mean, it's possible we haven't found the right reducible models, the right simpler representations that are useful, that are also fairly well biophysically informed. I mean, the representational models we use are kind of very loosely biophysically informed, but they're not. You don't have actually an explicit representation of neurons and conductivities in them, so they're kind of. Yeah, it's hard to sort of claim they're biophysical in that sense. But yeah, I would argue that no one's actually, in terms of this kind of like generative modelling of brain activity through biophysically informed models, there's not like a clear win yet in terms of it being delivering the kind of reducible computations that are really useful, like you see in physics and other domains. Hopefully that will change, and there's people working on it, and they might come up with something. Probably requires a bit of a paradigm shift from what's being done at the moment. But I guess we'll see.
Yeah, I guess it's interesting because biophysical contains physics, right? So it's all, there's an assumption there that the model itself has to be, it's more like modeling a physical phenomenon rather than the brain itself. I guess the bio as the brain part. Right? Yeah,
I mean, it's physics in as much that you've got, I mean, I don't know how much you've looked at the kind of models that are used in those kind of models of neuronal populations, but they are conductance-based models. They're basically like little mini electronic circuits. So that's physics. Yeah. I guess that's where perhaps to some extent why it's described as a.
But that's also a huge reduction, right? Of the complexity of.
Yeah, totally. Yeah. And there's non-linearities in there, of course. Hence where that's inspired artificial neural networks. But yeah, it's very simple compared to what the brain's really doing.
All right, let's zoom out a bit. What is the most interesting for you in this field, in neuroscience?
Right now, I just basically love playing with data. It's probably the most sort of simple answer to what I enjoy about this field, and it's a data rich field. So you can really get your hands on some messy data and work out how to make sense of it and find structure.
So what kind of data specifically?
Well, I work on brain imaging data, as you know. So it's that's the sort of data that does have quite high dimensionality, quite high amounts of spatial and temporal resolution, depending on which modality you're talking about. And then also you've got some variability quite a lot of variability over the population as well, over different subjects. So there's a lot to work with.
So just to be clear, this is things like fMRI, MEG,
yeah,
EEG. Can you briefly explain the differences between these?
So fMRI is probably more familiar to people. MRI machines, I guess people have seen those in their hospitals who've had like a brain scan for certain types of conditions, or maybe on the news. And those are measuring tissue properties through quantum mechanical sort of interactions. It sounds quite complex, but I guess the bottom line is that you can basically use a magnet, a very high field magnet, in combination with radio frequency signals to basically manipulate and then record the tissue properties inside the body. So that's used to form images of the brain, like you can see the difference between white matter and
three-dimensional
grey matter, three-dimensional images, and you can see the CSF and the bone and so on. So that's really good for looking for like lesions typically and other kind of anomalies in the brain, for example, or indeed in other organs. And then the functional MRI is a variant of that, where you use a particular manipulation of the quantum mechanical properties via the radio frequency signals that are kind of interacting with the tissue in the brain to basically look at a signal that's sensitive to the amount of blood, particularly oxygenated blood. So you basically end up measuring changes in oxygenated blood which occur close to the neural activity. Just turns out when the brain produces neural activity over populations of neurons, it needs to obviously provide energy for that, and that causes capillaries around that area of activity to kind of fill up with oxygenated blood. And it's that we detect in these MRI scanners. And it's got like a couple of seconds resolution in terms of the 1 or 2 seconds in terms of how quickly we take a picture of the whole brain, this 3D picture. But because we're measuring blood flow, it's even kind of slower than that, the information. The blood's got to flow into the capillaries around the neural activity and then flow out again. And that's going to typically be on the order of several seconds. So you end up, so you end up with quite poor temporal information. But you get pretty good, very good spatial information. Because even though you're measuring the capillaries, it's actually quite surprising how local in space those are, down to the kind of millimeter level.
Because the whole brain needs capillaries, right?
Yeah, it's very efficient in terms of where it increases the blood flow. Basically, capillaries dilate locally in ways that aren't fully understood.
So it's very focused.
It's very local, around where neural activity occurs. So that's where the spatial resolution comes from, even though it's blood flow.
That's kind of surprising the brain can do this, no? Or I guess the body as well. Okay.
Yeah, I'm out of touch with that sort of research, but last time I was in touch with it, I don't think that mechanism was fully understood. It's like a local signalling mechanism. There's when you get the assumption is, I think, that when you get some activity occurring, then there'll be a certain chemical signal that is sent out as well. I think nitrous oxide, I think, is one candidate, and that basically dilates the capillaries locally, and that causes this increase in blood flow to occur sort of naturally, based on the kind of what's available within the more local.
So it's a biochemical.
Yeah, so that's why it can be so focal, because it's kind of like this whatever this signalling chemical is, it's going to be limited spatially around where the neural activity is occurring. So that's fMRI. Obviously, I can say a lot more on that, but it's probably enough for now. So good spatial resolution, but pretty poor temporal resolution. Also, one thing I mentioned that's probably quite important is the vascular confound, in that you often are interested in looking at what happens between different conditions. So when someone's doing a task, or for clinical research, when someone's on or off a particular drug, or has a certain disease versus not having a disease. And quite often these things like drugs and disease can have a reasonable chance of affecting the vasculature just as much as the neuronal activity.
So it's hard to study.
And you can't tell which one's which purely from fMRI. If you get, when people have got dementia, you'll see a change in the signal you get from fMRI, and you won't know if it's caused by changes in the vasculature or changes in the neuronal activity. So that's a difficulty with fMRI as well. So then you've got MEG and EEG, which are more directly measuring the neuronal activity by picking up the electrical and magnetic fields that are travelling from the neural activity to the surface of the head, and they are, because it's literally picking up the electromagnetic transmission. It's going to be kind of instantaneous, and it's a more direct measure of the neuronal activity, albeit at the population level. So yeah, that's the main difference, and so very, very good temporal resolution, millisecond sort of temporal resolution in terms of recordings typically, but quite poor, relatively poor spatial information, because you don't know necessarily where these signals are coming from when you're recording them outside the head.
Yeah.
So EEG, people are probably familiar with, you've got electrodes placed on the surface of the scalp, and there may be typically sort of anywhere between 32 and 128 of these electrodes. And so you're trying to work out where the activity is coming from in the brain from quite a limited set of recordings. And on top of that, you've got all of the distortion, dispersion of the fields as the electrical activity moves from the source of the activity in the brain through all that different tissue, through the scalp, through the skull, to the electrodes. So you just don't know where it's coming from. MEG is a bit different in that it's measuring. There's no contact because the magnetic fields can move through air fairly well. So that's just basically sensors or coils that are held in an array around the head, like a helmet. And if the fields pass through those coils, you pick up a signal. But these fields, in terms of the magnetic fields, they're much less distorted and dispersed by the tissue it passes through, so the skull, the scalp, and so on. So you can much more reliably work out where the signal's coming from with MEG compared to EEG, which is why MEG is even done at all, to be honest, because it's quite expensive compared to EEG.
Yeah, it's much more expensive.
But it has much better spatial resolution, so you can resolve things down on the order of several millimeters, depending on where in the brain it is, a few millimeters in some cases. So it's better than EEG spatially, but not as good as fMRI.
But ultimately, they both measure like the electromagnetic. So one measures the electric part of the electromagnetic field, and the other measures the magnetic.
Yeah, exactly.
But there's also a question of which neurons exactly the signal comes from, right?
So that's. Do you mean in terms of the orientation of the neurons? Is that what you're alluding to?
Yeah. So EEG can capture some neurons, and MEG can capture.
So because the electromagnetic waves that you talked about, as they emanate from some current that's happening locally in some a small group of neurons in a little neuronal population, the fields go off in different directions. So that gives you a different kind of sensitivity for the two different devices, depending on the orientation of the neurons in the cortex.
Yeah.
So they can provide complementary information for that reason, as well as complementary information in terms of how they get distorted and dispersed by the tissue they pass through. So you can do simultaneous EEG and MEG. In fact, we do some experiments here at OBAR that do that.
So you've been doing a lot of MEG recently. Why is MEG in particular cool? And how does it influence the type of questions we can ask?
So I did fMRI for like 10 years. I still do fMRI now, just not as much. And I switched to MEG mostly through a job opportunity, if I'm honest, in the first instance. But I also was excited to work with some what I think is richer, more interesting data than fMRI. fMRI is so simple temporally, it's a bit boring. Whereas MEG is kind of almost like overly rich temporally, but there's enough spatial sort of specificity that you can get your hands on stuff that's useful to work with, both spatially and temporally. So yeah, like an exciting new domain. You start worrying about oscillations and oscillatory activity and how that interacts across different frequencies, what information is carried in those oscillations, how that relates to what we can't measure so easily in MEG and EG, which is the spiking of neurons, but you can do studies that help you to bridge that gap. Mm-hmm. And then, of course, you also try and link back to fMRI as well. So it's a really sort of nice, quite a nice bridging technology between more kind of invasive techniques for recording brain activity, where you can also record firing rates of neurons, through to the cruder spatial information you get across the whole brain in fMRI. So yeah, quite a cool sort of niche to work on, exciting data in terms of its richness, and it allows me to play with the kind of machine learning techniques that I've always used. Was that? I forget what the question was. Is that
yeah,
right?
I guess, yeah, I was wondering specifically MAG, how does it influence what kind of research you can do compared to fMRI or compared to invasive stuff?
Yeah, so MEG and EG, as I've alluded to earlier on, it's that temporal insight that you can get. So people use it when they have questions about the timing of events.
Yeah.
In particular, MEG then is particular technique that's useful when you care also a bit about what's happening spatially and also the timing of the events, because you want to kind of know about both to really understand the system better, or how it might go wrong in disease. So that's where MEG is useful. And then also, I talked about this vascular confound issue. So in disease research, like we do some research on Alzheimer's here. That's a case where you want to get some confidence about what you're measuring as being neuronal rather than vascular. And we also collaborate with drug companies, again, who care about if their drugs are acting on the vasculature or if they're getting through the blood-brain barrier and acting on the neuronal activity. So again, MEG's very useful for that as well. So yeah, that combination of free of vascular confounds, and when you care about understanding the timing of events, and you want to get insights into timing of events while still maintaining some spatial information, that's kind of the sweet spot that MEG provides.
So would you say the cost of MEG or these benefits outweigh the cost of MEG at the moment? Or
I mean, if there's certain types of research you arguably can't do very well with it, or at all. So yeah, in those situations, it's worth doing. Yeah, absolutely. But I guess it's something to always keep an eye on with technologies. But I think it's been proven to be providing a sort of unique service, which is serving the neuroscience community.
Yeah, providing this trade-off
that you couldn't get via other approaches.
Yeah. Shifting gears a bit, I was wondering, how did you become interested in neuroimaging, and what led you to pursue this career in Oxford?
That's taken me way back. Do you want my entire life story?
Whatever you feel is
I was born in 1990.
No, I guess what influenced you to pursue neuroimaging in an academic sense?
Yeah, it was a little bit of a it wasn't like a purposeful journey at the early stage. So yeah, I did engineering as my degree in Oxford, and specialised. It's a general engineering degree, which I thought was brilliant. Doing general engineering rather than having to specialise. And then, obviously, towards the end, you start to specialise once you know what you're doing. So I specialised in information and electronic engineering, and took some fourth-year courses on things like neural networks. This is back in the mid-90s, neural networks, and multi-layer perceptrons, backpropagation. So it was around then, but using very niche applications. And also a course on biomedical engineering, so those sort of two together really, I know, quite like those, they piqued my interest. But then, despite that, I had no plans to be an academic, so I went off to work for IBM for 3 years in London, working on financial accounting software and as a technical management consultant. It was good fun, but I got a bit bored I wasn't motivated by it and missed the intellectual challenge. So got in touch with one of my old lecturers from my 4th year undergrad, and a guy called Mike Brady, and he worked in basically the robotics department in engineering, but did a lot of biomedical engineering as well. So yeah, I got in touch with him and signed up for a PhD, or DPhil, as it's called in Oxford sometimes. And yeah, just basically in biomedical engineering, working in his group. And he worked in a whole bunch of different things. So when I signed up, I didn't know what I was really going to specifically work on. There was possible work in X-ray mammography, and then also in the brain stuff, as I called it back then. So I actually worked on both for the first sort of, I don't know, month or two. And I went up to meet Steve Smith up at FMRIB, the fMRI of the Brain Centre here in Oxford. And so I ended up doing my DPhil with him, and obviously still with Mike, split between engineering and the fMRI Centre in Oxford.
So you didn't really have
that was kind of my journey, and it was. Yeah. So I guess the question was about why did I choose neuroimaging? So why did I choose that and not X-ray mammography? Well, it just seemed like there was more scope for asking interesting questions and doing. It felt like there was this like huge expanse ahead of where I was going to come in at, of things that could be done and could be possible. So I kind of had a sense of the richness of the data, and also it was very new. The fMRI Centre had just opened then, so this is the start of a exciting new venture. I could sort of sense the sort of the sort of thrill of this new thing starting, and the possibilities being who knows what. So I jumped on that bandwagon at that point and sort of stayed on it ever since.
So you didn't really have any neuroscience training or background?
So again,
You didn't really have any neuroscience.
Oh no, I was pure engineer at that point.
Yeah, that's kind of similar to what happened to me.
Yeah,
engineering, computer science, and then the DPhil. Yeah.
Yeah, you're kind of looking for. I mean, with engineering, it's obviously applied science at the end of the day. So you've got to find an interesting application, I think.
To do research.
Yeah, to do the engineering stuff on. And neuroscience is a very vibrant area, multidisciplinary. So it can be and it's an interesting area that we can all kind of relate to. And there's also for me, I guess for you as well, there's an interesting interaction in terms of artificial neural networks and artificial intelligence as well, that kind of dovetails or interplays with neuroscience as well, which is kind of an interesting extra perspective. And
so then during your DPhil, you did lots of fMRI?
Yeah, it's all fMRI, 100% fMRI.
What was your topic on, or thesis on?
I think my title of my thesis was something like fMRI time series analysis.
Quite generic.
Yeah, it was time series.
Yeah. But that's when these methods were actually developed.
Yeah, right? yeah, exactly. Yeah, people didn't know how to analyze fMRI data, and the thing that I didn't enjoy so much was the kind of hardcore stats, and that's still something you have to contend with to this day. But I see it as a kind of necessary, not evil, that's overstating it, but something you have to do, not something I enjoy doing. When you have to worry about p-values, particularly in the classic stats framework, which is where I was working in my DPhil, and still to this day, quite a lot of neuroimaging science is done using classical null hypothesis testing. And I just found that quite hard work. Mm-hmm. But I discovered Bayes in my DPhil, and that was a big moment. That was a major turning point. That was quite a seminal thing for me to discover Bayes. I can't overstate it really. I can talk more about that if you want, but that was. Yeah,
we'll go actually into that a bit later. I was wondering though, how do you think the field has evolved since you began your work, or since your PhD?
The fMRI field or the neuroimaging field in general?
fMRI or neuroimaging more in general. Yeah.
It's kind of hard to sort of separate my own journey from what the field's doing. It's kind of. Because obviously for me it feels like things were so focused on MRI in those days that one version of the answer to your question is that things have opened up with more modalities being considered, and the mix of information you can get being really important from those different modalities. But that much is because I've been on that journey myself. So yeah, it's kind of, I guess, the biggest change in the early years that was kind of, I guess, more than the fMRI world was going from understanding the brain as kind of trying to basically map which brain areas activate when someone's doing a particular task or being subject to a particular stimulus. So-called blobology, as we sometimes call it. So you have like maps of blobs of activity in the visual cortex at the back of the head when someone's looking at a flashing image on a video screen. And so you might then come up with another task or get someone to tap their fingers, which part of the brain lights up, where's the blob, when people are tapping their fingers. And I'm oversimplifying it, but that was the basis of a lot of the early stuff. And then one of the big changes has been moving from that kind of sort of segregated perspective to a more integrated perspective where you're looking at networks of brain areas interacting. And there's not just one brain area that's responsible for doing X or Y, it's interactions between multiple brain areas that kind of bring the overall behavior of the brain. Mm-hmm. From the analysis point of view, the computational modeling point of view, it was switching from kind of sort of simple time series analysis techniques, signal processing, to kind of techniques that can work with networks and compute network interactions, and then similarly doing the stats on switching the statistics over from one to the other, and switching the modeling from one to the other.
And you've been a major driver in developing these core methods for.
I've worked more on networks since I moved into electrophysiology, into MEG and EEG. I would not claim to be one of the early drivers in the fMRI world of switching over to networks. Once it became clear, it was an important perspective, and around about then I was switching over to electrophysiology, so mm-hmm. It was a natural thing to start working on in the MEG EEG world instead.
I guess interaction means you need to study that. You need to take into account time, right? So it makes more sense to study it in electrophysiology.
Exactly. Yeah. So one of the big questions is when you start looking at networks and how brain areas are interacting, it's like, well, what do you regard as being a significant or a measure of a certain pair of brain areas interacting? Mm-hmm. How can you tell when they're interacting? What do you find in the data that tells you they're interacting? And in electrophysiology, that question is kind of richer. There's a lot more ways in which you think about the signals from two brain areas telling you the brain areas are interacting. Whereas with fMRI, it's a bit simpler because of this loss of temporal information I talked about earlier.
Yeah.
So in fMRI, you just use simple correlation typically. So the two brain areas fluctuate sort of at the same time in the same way to some extent, which you can measure with correlation.
Yeah.
So it's kind of boring, as I mentioned earlier. And so a lot of the work in fMRI is more done once you've doing the stats on that kind of thing, or once you've computed the network, trying to come up with useful summary measures of the network, sort of graph theoretical approaches, which people get carried away with. But I think that's less interesting than trying to understand the kind of rich interactions that occur at very fast timescales. At the sort of timescales the brain's going to be doing stuff, like this 10, 20, 50 millisecond timescale. How our brain areas interacting, and how does that relate to very fast cognition as well, at the same sort of timescales, which you can't do with fMRI, but you can do with MEG, and to some extent with EEG.
Yeah, it's closer to the biophysical
yeah.
reality underlying it. And thinking about machine learning, what are some discoveries that you've seen that were made possible with machine learning in neuroimaging?
Interesting question. Yeah, so just to carry on from the previous theme, the kind of network perspective, people were just initially doing it with these straightforward correlation approaches. So I think the very earliest example of that was a study by Bharat Biswal, where he basically took the time series of signal from like an area of the left motor cortex, so the area of the brain that's going to be involved in producing a movement, and he took that time series, that signal, and correlated it with the whole of the rest of the brain, and found that as we now know straightforwardly, the right motor cortex, so the left motor cortex was the signal, kind of the reference signal, correlate with the rest of the brain, and the right motor cortex shows a high correlation. with the left motor cortex. So it pops out as this kind of blob, right motor cortex blob, showing the areas of the brain that are correlated with the left motor cortex. And that was kind of almost like the first network analysis, you could argue, of a sort that was done. And then so people would then put these seeds all over the brain. So like stick what we call the seed reference signal, stick a seed in the visual cortex and find which areas of the brain are correlated with the visual cortex. And you see the visual system and aspects of the sort of dorsal and ventral stream, depending on if they're doing a task or not. So yeah, that's the kind of state of the first wave of network analyses. But one of the problems with that is that you don't, you have to choose your seed voxel. And how do you summarize the networks that are kind of happening without having to choose a seed voxel? And that's where some of the machine learning techniques sort of first came in. So this is like unsupervised learning approaches like independent component analysis. And that had a big role to play in the early network analysis that was done. It's unsupervised. You make some quite minimal assumptions about how to unmix what's happening spatially, what's happening temporally, and you can essentially pull out brain areas that are fluctuating together, correlated with each other, without having to choose a seed region. So that was a very popular machine learning technique in the early stages. Other machine learning would be when you then come up with, you start trying to do cleverer modelling of what's happening temporally, or over subjects, or over space. These are like the kind of the early attempts at generative modelling of the signals. And then the machine learning that's done there is Bayesian inference. So how do you extract in terms of probabilistic representation of what's happening spatially and temporally, which brain areas are doing what and when. Bayes was that's kind of how I got into Bayes at that stage. It was like trying to fit more complex, cleverer models to the data than the bog standard sort of classical stats that people were doing. And then being probabilistic, so making sure you weren't just finding things happening just by chance, just because of the random fluctuations in the data, and therefore random fluctuations in your model parameters. Yeah. And Bayes was a really good way of doing that. So I collaborated with a guy in engineering in the pattern analysis research group called Steve Roberts, and he introduced me to variational Bayes, and I was doing MCMC as well, Markov chain Monte Carlo, and these are all ways of basically using Bayes rule to infer on a generative model. So you've got a generative model that generates your data, but of course in practice you would get the data and you want to infer the model parameters from the data. And so inverting that generative model is what Bayes can do for you. Can't do it mathematically, analytically, on pen and paper for most realistic cases. And so it uses numerical techniques or approximations. And MCMC is a pure numerical technique, like brute force, but still quite clever. And then variational Bayes is kind of a combination of the two. You do some pen and paper maths, but then you use the compute to iterate between updates to your model parameters. But at the end of the day, you get this probabilistic estimate of your model parameters. What's the expected value, what's the variability around that expected value, given the evidence in the data. So it's a really powerful framework to use, which is why I latched onto it in that, and towards the end of my DPhil. So that was kind of a machine learning-y thing, I would argue. So those are the two obvious ones that spring to mind in those early days.
Yeah, it's interesting you mentioned ICA, which is such a basic method nowadays. And but at the time it was very seminal, right?
Yeah, it was hardcore at the time. Yeah, it was like, I remember spending many a reading group trying to work out how it works, the magic of it and what it's doing, what it assumes, what those assumptions mean, the different versions of ICA that were around, how do they compare, how do they relate. And actually it all fell into place when I found the sort of, I think the first version of Bayesian ICA. It's like, oh, okay, that's the generative model. That's the assumptions. That's the beauty of Bayes, even for things like ICA. It's like a non-Bayesian perspective is like almost like this sort of sequence of steps that you do with some assumptions that are sort of scattered throughout it. Whereas with the Bayesian approach, you got to specify your mathematical model, your generative model, and then you've got this equation, Bayes rule, that tells you what to do with it. And it's just like there's no room for hiding behind nonsense or kind of waffle or like sleights of hand. It's just like that's it.
You
And
still
so-
need a good amount of data for it to work, right? The
Not-
base approach.
Not always.
Or does it work with
No, because
relatively little data?
it can do, yeah, it can do. In fact, it can work better than other approaches in some cases because it can handle the uncertainty. If you do like point estimation Bayes, then it's going to struggle. But if you're doing probabilistic Bayes, where you're taking into account uncertainty in parameters, which is if you're doing Bayes properly, that's what you should do. And the reason you don't do it is because you're either lazy or it's computationally too difficult, or it's just not worth the hassle because the uncertainty isn't big enough in the ways that you care about. But otherwise, in principle, the uncertainty is there to be had from Bayes, and so you can therefore cope in principle with small amounts of data, and you'll see your uncertainty increase because you've got less data.
So what this gives you is a generative model of the data.
Bayes doesn't give you generative models. I would say it's the way of inverting the generative model. The generative model is something you can come up with sort of separately, if you like, and then you can then invert it with Bayes rule.
So but then it helps you learn that model.
Yeah, exactly.
Yeah, but then there's a question of what do you do with that model, because that doesn't really tell you anything about the brain, right?
Well, it depends what the model is.
And I guess just to zoom in a bit on your work, a major theme has been in compressing the rich dynamics of, for example, MEG data into, for example, a core sequence of spatially distinct states.
Yeah.
So I guess you've been using Bayes for this.
Yeah, that's right. Yeah, so that's a good example of the generative model Bayesian combination that we talked about just now. And it's the generative model we've used quite a lot, as you know, is the hidden Markov model, and that's particularly. But it sort of came to me as a good way to do things on electrophysiological data, because we have such good temporal resolution, and the assumption of there being mutually exclusive states seemed to be more useful when things weren't being temporally blurred, like in fMRI data. Although we actually use it on fMRI data as well, to some reasonable success, despite that. So yes, I mean, I think about HMM sometimes as being like a sort of augmentation of ICA. If you bear with me on that.
Not sure, but come on.
Well, it's in terms of the progression of the thing. So we were using ICA. Actually, when I first started working on MEG, collaborating with Matt Brooks on using ICA on MEG for the first time, hadn't really been done with success. And so given what we've been doing in fMRI, we worked on the MEG application of ICA, and it worked pretty well. But actually we had to throw away some of the temporal resolution to make it work. It didn't work when we tried to maintain the high temporal resolution. Whereas the hidden Markov model was able to maintain a high temporal resolution because it was kind of cleverer modelling of what's happening when a particular state activates. So ICA is just certainly in its form that it was back then, standard ICA. Each component is kind of analogous to the states we look at in the hidden Markov model.
Oh yeah.
I'm jumping in quite deep here quite quickly, aren't I? Do you want me to backtrack?
No, that's okay.
So in both cases, your HMM and ICA, you're taking data over multiple brain regions, or sometimes called channels, and you've got a time course for each brain region or channel. That's your kind of data. We're going to try and map that into a representation where you're explaining that data using a certain number of networks or states or components. So networks is how we describe it if we're talking about the brain. Components is how you describe them if you're talking about ICA, and states is how you'd call it if you're talking about standard HMM approach, hidden Markov model approach. But they're all in principle the same thing. They're describing mapping from the number of brain regions down to this a smaller set of networks or states or components. So it's like a compressed representation, as you said. So in that representation, you've got these different stage channels or networks, but you've also got how those vary over time and what's the dynamics of those networks. we'll just use the word networks from now on. So what's the dynamics of those networks? And also what is the spatial configuration of those networks, what brain areas are involved in those networks. So in ICA, the spatial configuration is just a spatial map in classic ICA. And the time course is like a continually varying time course for each component. So that's quite rich. But it hasn't got much in the way of regularization in it. It's like pretty free to be what it wants. Unless you make the assumption of temporal independence, which we'll ignore for the minute. But it's, generally speaking, it's pretty free to sort of fluctuate around. So there's not much to constrain it or regularize it. And that's the reason why I think it didn't work very well on MEG data, as well as the poorer spatial description that you've got, which is a spatial map. Whereas with the hidden Markov model, it's a slightly more complex model where your dynamics for each network is a binary time course, which tells you when a particular network is activating. And only one network can activate at any one point in time, so it's quite a strong constraint, but that really helps you define stuff that's identifiable in the data. And then spatially, we actually explicitly are modeling networks, not just spatial maps. It's like which brain areas are actually correlated with each other, interacting with each other when a particular state or network switches on. So it's richer in that sense as well. So yeah, that's a kind of a very brief hand-wavy description of the difference between ICA and the HMM. And the HMM, through its better regularization temporally and through its richer spatial model, worked well on HMM. Worked well
MEG.
on MEG data, and revealed this very fast switching, like this 50, 100 millisecond timescale, the sort of timescales you care about with cognition. And you can see it showing task dependencies at these fast timescales. And no one had seen, arguably at the time, resting state networks, which we haven't talked about, but it's one of those things that studied quite a lot, like the resting state of the brain. It showed those networks fluctuating on again, these very fast timescales, much faster than had been looked at previously with fMRI.
So what happens in the brain when nothing is going outside?
Right? I knew you were going to ask me that. So this is the resting state. So this is the resting state. You have somebody lying in the scanner doing, like you say, nothing, ostensibly, whether it's an MEG or an fMRI scanner. But the brain isn't doing nothing. It's doing all sorts. It's fluctuating very, very strongly. In fact, if you look at the extent to which the brain additionally fluctuates and they're doing a task that you're in control of in your experiment, it's kind of quite minuscule compared to the overall ongoing activity that's happening anyway. The brain's just doing as and of its own accord. It's like, what does that, why is it doing that? What does that mean? Well, that's one of the interesting research questions from the last sort of 20 years that's not been fully answered to this date.
Well, I was going to ask you, what have you found out about what's going on in the brain?
What on earth is all this? What is the brain doing? Wasting all this energy doing all this stuff?
Because on the method side, you can characterize it with networks and the dynamics of.
Well, even that wasn't obvious though. I mean, like when I first started, it was treated like just noise.
Yeah, so that's a
we literally
nice
thought
result.
it was just like noise, coloured noise, but there's noise.
Really, people didn't think there's anything
until this Barrett Biswell study came along that I mentioned. People thought that the kind of what was called the temporal autocorrelations, the spatial temporal correlations that were happening above and beyond the task, they thought it was just kind of noise, physiological noise, if you like, maybe just that fluctuations in respiration and blood flow, and maybe, I mean, no one really thought about it being meaningful neuronal activity. I mean, it seems obvious now that that was a big thing to overlook.
Yeah,
and maybe I'm exaggerating. Maybe other people.
I'm sure people have noticed because there is. You need to maintain consciousness, right? Like that's an important function of the brain, and you think about memories, and you need to store memories and all this background processing.
I mean, obviously, we now both know that and both agree. But maybe this reflects my naivety because I was quite new to the whole thing at that stage. So maybe it's my own naive perspective and arguably the naivety of other people in the field at the time as well, because the brain hadn't been studied that much at the whole brain level, non-invasively in humans. You had understanding of the brain to some extent through EEG in terms of these things like alpha oscillations, but those weren't necessarily regarded to be cognitively meaningful, other than maybe just like markers of crude brain state, but not like necessarily
like focus.
Attention. Eyes open versus eyes shut, that kind of thing. Yeah, people. I mean, I think it wasn't just me. I'm sure there were cleverer people with more foresight who were thinking about it being neuronal earlier than I realised. But there was, I mean, for example, there was a lot of pushback when people started claiming it was neuronal. People saying, "Oh, it's just you're just picking up respiratory cycles and heart rate and variability."
Yeah, but I guess that's because it's hard to show, statistically these kind of things, like concretely that it's above.
Yeah, exactly. Yeah, but it's almost as if the default idea was, there's nothing neuronal in it. You have to prove to me that it's, rather than the other way around, which is like you can almost like now it's like, well, prove to me it's not neuronal almost.
Yeah. So like the null hypothesis would have been the other.
Yeah, in a way, yeah. It's just because of the mentality of the field at the time. But yeah, so now we know it's neuronal. You have to take into account some of the physiological confounds like respiratory and heart rhythms. But once you do that, the stuff that's left over is clearly neuronal. So what's it all about? You've already mentioned a few things when you were replying to me before, like things like the thing that we've worked on is looking at memory replay. So when someone's doing a task, your way of doing it, you expect brain activity when you're doing a task. So for example, you might be learning, the classic experiment we do, might as well stick to that to keep it simple, is you look at sequences of visual images. This is work done by Tim Behrens in Oxford and at UCL. You look at a sequence of pictures on a video screen, basically, and you learn the sequence of pictures that's going to be happening. There's different sequences, and you're learning those as you do the task. And you can also, whilst people are looking at the pictures, you can actually train a brain decoder to tell you when somebody's brain is activating in response to that picture, or is activating the representation of that picture more generally. So that's kind of part one of the experiment. And then you stick somebody in the scanner doing this resting state thing that we talked about. So they're lying in the scanner doing absolutely nothing in particular, or sitting in the scanner in the case of Meg, doing absolutely nothing in particular. And you'll see the brain patterns that were happening when they're doing the task spontaneously reactivating. But they will do so in the sequence of the visual images that have been learned previously. So it's not just like random fluctuations of the visual images themselves, which is kind of cool anyway. It's also richer, cognitively important information, like the sequence of the pictures that you've been learning.
So I guess that's something learning specific that the brain does.
Yeah, so it's like, so you obviously when you were doing the task, your brain was learning. It was learning to kind of recognize when there was a particular sequence, and it was strengthening the connections in the brain that help it to know that that's a sequence and what the sequence is, so it can make use of that information in the future. But turns out that's not enough. You also want to redo that whole thing, and one would assume, and there's evidence to show this, that it's to enable the brain to strengthen the connections, make the memory laid down more strongly, and also to augment it and make it more robust in the ways that we do sometimes in artificial neural networks.
Like variant.
Yeah, exactly. Or injecting noise, whatever it might be. So it's basically doing that. It's offline from the task. It's strengthening and laying down the memory. There's also possibly transferring it from certain brain areas to other brain areas as well, is potentially part of for longer term sort of storage, to give it a very crude, hand wavy description.
Storage. I guess nobody knows how memory works.
Yeah, so
we should.
Very interesting. I mean, I think there's experts in memory who know more than I do for sure. But even they don't know fully how it all works. But there's certainly strong evidence for this replay phenomenon, and understanding that replay phenomenon is an important area of basic neuroscience research to understand how memories are laid down and when that can go wrong in disease as well.
Yeah, that's quite interesting, because I guess that's a general thing that happens with learning. Like in theory, you could show that with any task. For example, a motor. I don't know if this has been done with.
I know it's been tried.
Motor tasks.
Yeah, it's been tried. I know. I didn't. Last time I heard it, last time I looked, it hadn't been published, but it's been tried and met with motor tasks as well. Mm-hmm. Yeah.
Or
So
playing music, for example.
I think once these initial replay, so originally the replay experiments that I described were done in animals and invasive recordings, and there's a huge literature in mice, like following routes around mazes, and you see these things called play cells and hippocampus firing in sequences when they move through the maze, and then you see the same sequence happening when the mouse is at rest. And I should say it's not just rest, it's also sleep. I mean, sleep, in fact, it's even stronger in sleep.
So that's something we share with the mice, this kind of...
We don't remember mazes in the fMRI scanner, although there are some virtual reality experiments that are being attempted where you do that. Instead, we have these sequence of visual images in the first pass of this. But now people are trying all sorts of different variants on it. It's a kind of slightly new field that's taken off the last sort of 2 or 3 years. But yes, that's one example of what we know happens in the resting state. I mean, other than that, there's obviously our ongoing mind wandering. You have, if you try and sit or lie still with your eyes shut or open, thoughts pop into your head. I mean, we know that happens. There's a need to be, when you're awake, there's a need to be ready to go at any point in time. So your brain can't just be switched off. It needs to be ready to go. So it needs to be kind of in this state of readiness. And so a lot of the ongoing dynamics, the activity, is about being in a state of readiness. So as soon as something comes in, you can respond to it quickly.
So it's like this dynamical system, which is as soon as there's a perturbation.
It's regarded as being the dynamic repertoire of the brain is one of the fancy phrases that I've heard to describe this ongoing activity that is almost like some people have argued through the use of biophysical models actually that you naturally get this kind of volatility, this fluctuations when you have a system that is on this sort of state of readiness, where you've got this balance between excitation and inhibition, such that you can release the inhibition and immediately something can happen very quickly.
So sort of from the structure or how the brain is wired.
Yeah, so it's a natural state of the brain. If you have this balance between excitation and inhibition, and you want to be in a position to be able to like release inhibition and go really quickly, it's kind of like almost like a natural consequence that you'd have fluctuations that are ongoing. I don't think it's as simple as that. I think obviously the brain is then making use of those fluctuations and doing meaningful tasks like consolidating memories and motor activity, and probably doing obviously useful conscious stuff as well, like mind wandering, pondering on stuff, which sometimes can be destructive, but there must be an evolutionary reason why we do that. So it's obviously overall worth it, and probably other stuff that I haven't thought of and haven't mentioned yet in this chat.
Isn't there this theory that the brain is always trying to predict the future, like predictive modeling? Yeah, and that's how all its functions sort of arise, is it?
That's a good point.
Trying to predict, right?
Yeah, so this state of readiness I was talking about is like, it's kind of its ability to react quickly is also because it's constantly making predictions. Yeah,
so it has like a model of the
world. It's got to be activating in a coordinated way in order to be able to do that. So that's I think that's a good sort of additional perspective on it as well.
But it's quite hard to study. I feel like there hasn't been any concrete where they link brain activity with how it tries to predict.
Well, I mean, so people. I mean, you can. There are studies. It's not my area of expertise by any stretch, but there are studies that will, try and predict what you're going to do based on the state of your brain activity in ways that you're not aware of as the conscious person. And there's quite a few studies that show that your brain is doing stuff that is kind of able to predict what you're going to do where you consciously aren't able to do that. So that's an example of, I guess, of the brain. It's predicting what it's going to do at least. Yeah. And part of that will be also folded into that will be the brain trying to predict what's going to happen around it as well in terms of other people and the environment, inevitably.
Yeah, so there's all this sort of interesting interaction with the chemical part as well, because, so for example, dopamine gets released right when there's a mismatch between prediction and
it's a learning signal. Absolutely. Both when things go wrong and things go right, you get a differential dopamine signal to help you learn from negative and positive feedback.
But yeah, in terms of your methods you've been developing, I guess they sort of sit at this intersection of explainability, so saying things about the brain, but then also modeling it and building this kind of models, and also both in healthy people and people who suffer from diseases. So I guess we've already talked a bit about this, but how do you achieve designing such methods that are directed towards explainability?
How do you achieve models that have explainability?
Yeah. What are some core ingredients that you used? Or
so it's kind of finding that sweet spot where you I keep using that word sweet spot, don't I? Don't know why. It's finding that perfect area where you're kind of able to interpret what's happening, what you're finding out from the model in a way that's useful and is kind of, if not biophysically accurate, is sort of has got some relation to location in the brain and times and when things happen in the brain. So that knowing where things happen and when things happen is kind of useful information. Whilst not being biophysical, it's a useful representation that then can be linked to other things that are happening, at the same time, or when there's disease in a certain brain area, that kind of thing. So I think models at the very least give you some insight into when and where things are happening, and a network perspective, which brain areas are interacting together to make something happen. So that's kind of like this level of description that I've kind of gone for, where you're getting insight, but there's nothing biophysically in that. I'm not constraining it based on which brain areas are connected by the white matter. I'm not constraining the dynamics, via refractory periods or whatever it might be, or the synapse types that you have in certain brain areas and their time constants and so on.
You're letting the data speak for itself.
Yeah. So and I think that's obviously, I'm not claiming that's the only sweet spot to work on, but it seems one where we've been able to get models working in a way that's reproducible and finds interesting structure, that then relates to things that people are interested in. So, when people do tasks or perturb the brain or look for things like replay, or when things are then affected in disease, these sorts of representations have parameters in them that you can see changing with disease progression and drug interactions. So it's a useful representation. But there could be other levels of description that I'm sure that obviously there are, and other people work on those as well, which is great, and that's the diversity you need in a field like this.
But in general, it seems like compression is an important part of it, because MEG already contains both spatial and temporal information, right? But just looking at it, you're not going to be able to tell.
Yeah, you need
what's going on. So that's why you need
to extract stuff that's reliably repeatable. That's not just noise fluctuations. So compression in that sense, denoising at the very least. Yeah.
So in some ways, this is driven by our limitations of understanding high-dimensional data, because that's what MEG is, right?
Yeah. So
let's just say, in hypothetically, like humans would be able to look at an MEG time series and just understand what's happening.
Yeah.
But because we don't, we have to build these compression models.
Yeah. You're right. It's a summary of what's happening in the data. Summary equals compression. So that's true. Even so, what you say is kind of valid in that the human brain and the way you try and write papers, therefore scientific papers, they work better with a sort of simpler story. The human brain can cope better, and you can then write a narrative around that and explore things, and it limits the scope of possibilities as well, so it's beneficial pragmatically. So we end up working with models that have got quite a low number of networks or states or components that perhaps we actually could work with. It's not just a question of reproducibility, but it turns out that things are also most reproducible with MEG in particular when we work with a number of networks on the order of, I don't know, between 6 and 18, roughly speaking, networks. That's where we get good reproducibility, and it's also turns out quite a good level of complexity that the humans are quite good at talking about and working with and dealing with.
Another sweet spot.
Another sweet spot. But yeah, no, I mean, I'm quite excited about working on bigger data, and just seeing that. I expect that we'll see reproducibility happening with more complex representations, with less compression, if you like, of the. I guess it's still proportionally the same amount of compression of the amount of data.
Yeah.
But you'll end up with a richer description with more networks, more states, because you've got more information from more data to feed that. But that remains to be seen. But there's early suggestions that will be the case. So we're excited about that direction.
I always wonder what kind of phenomena or deeper understanding could we be missing out because of this compression. Because MEG is already like a huge compression on the neuronal activity that's going on inside the brain, and then we're compressing it even further to a number of states that you can count on two hands. Yeah, no, you're absolutely right. And we want to do better than that, for sure. Totally agree. But I would argue that is doing better than what we were doing previously, which is where we average the hell out of it temporally and spatially, which is a different kind of a dumbass compression, where we just average things over repeated trials in an experiment, or we average over space, or we look at kind of this mixed average in sensors. So that's a much cruder, less intelligent compression than what the things like the HMM give you. You're finding a much more intelligent, adapted to the data compression, and it is scalable. So we can look for richer, less compressed representations with the HMM and the newer techniques that we're developing. So there's scope for doing that. And as I say, with bigger data, that's the plan. So analogously, from moving from older methods to HMM, do you have a sense of what we may be missing out currently? What, as in like extrapolating from what we gain from the HMM and from moving from the old techniques to that? Is that what you mean? Yeah, with our current techniques, what may be missing? Like something that is in the MEG data, but we're not able to access it. I mean, the boring answer is just more spatial and temporal detail, and an understanding of the variability over subjects, population variability of those things. And that does sound quite boring, but it's actually, ultimately the sort of raison d'etre of neuroimaging. And then it obviously, that information feeds into new insights into cognition and disease. So that's kind of the very high level answer. You probably asked something a bit more neuroscientific than that, wouldn't you? Well, I mean, it's hard to predict, I guess. Yeah. I mean, if I knew that, then you wouldn't be. I'd be a better scientist. I'd be a better scientist than I am, or a clairvoyant, one of the two. How do you apply this kind of methods and explainability in clinical research? Because that's also part of your work, right? Yeah, so we take these techniques, and I mentioned this earlier on. You can take measures from these biophysical or these generative models, I should say, and those parameters, you compute them separately for each subject, or separately for each session of a subject, if you like a drug study, for example, like with or without drug. And then you look to see how well those parameters correlate with the change in drug or the change in disease over sessions and subjects in your study. So we look at, for example, things like the amount of time that a particular brain network or brain state is activated, or the rate of switching between brain networks, or the amount of time a brain network switches on for before it switches off again. So it's kind of like its lifetime or dwell time. So those are the kind of metrics we can feed into as features for doing prediction of disease and of drug states and other clinical states. So that's kind of the method that we use. It does provide useful biomarkers which are able to predict diseases that we work on, like Alzheimer's and motor neuron disease, MS,
Parkinson's, just to name a few,
So
epileptic seizures or intellectual spiking.
Yeah.
So yeah, it's able to, the features we get from this kind of description, this dynamic network description, you see changes in the features from those dynamics in these sorts of diseases.
So it's often about earlier prediction of these diseases.
Yeah, so so why is that useful? Is I guess what you're asking. Yeah, so why is it useful to have this link between the features you can measure in brain activity and disease or disease progression? Well, yeah, a number of different applications. One is early diagnosis. Another is stratification of subpopulations in a disease. So you might be certain types of disease, for example, certain types of AD. Need different treatment to other types of AD. So if you can stratify them using this kind of information, that's useful. The drug companies are interested because they want to know if their drugs are working. So if you've got a measure that's sensitive to the rate of progression of a disease, so if you, for example, in a study where people with Alzheimer's, you measure them at sort of time point 1, and then you measure them a year later, and you're trying to see if these dynamic network measures are sensitive to the change in degree in disease over the course of a year. If it is, then it's a measure that can be used to measure the change in the disease progression when a drug's being used. So the efficacy of a therapeutic. So that's the another application. It's used. People use it for pre-surgical planning. Not so much the HMM, but like information about where things happen in the brain. And that's actually the HMMs we're using for epilepsy diagnosis and locating the source of intellectual spikes and therefore epileptic seizures using MEG and EEG via the HMM. And that's kind of, it's not being used in earnest yet, but people are trying to see if that's a possibility for improving the planning of where to do the surgery in people with epilepsy. So yeah, there's a bunch of different things that gets useful. We're not kind of at the point where it's being used on the kind of in primary care or in the kind of really at the coalface of clinical work, but it's heavily involved in clinical research. Probably the fair summary.
With all your interpretability methods, can you speak to how they tie into the more general machine learning? So like the interpretability methods that are used in machine learning in general. Yeah. So how has that informed your approaches?
When you say what sort of interpretability, so I know we're on the same page, what sort of interpretability method for machine learning do you meet?
I guess interpretability in machine learning is often about figuring out how the model works, the machine learning model, whereas in your case it's more about how the brain works. So, but still there could be some overlap between those two.
And do you mean, and are you talking about the deep learning sort of version of machine learning?
Yeah, deep learning,
because that's where the sort of interpretability challenge really kicks in.
Yeah, because the complexity of the models is larger.
Yeah, no, that's an interesting question. It's kind of at the, as you know, it's kind of at the core of sort of things that we're wrestling with and trying to do with this, with the deep learning techniques that have come in the last few years, and in relation to what we've done historically with things like the HMM and generative models. So my take on all that, if I can gather my thoughts, my take on all that is that it's about identifiability. That's kind of the key concept that I would focus on here. So when we work with models that we want to be able to interpret or make use of, so if it's a biophysical model, it might be the strength of certain connections in a mean field neuronal population model. In the HMM, it might be the rate of occurrence of a certain brain network state. We want those parameters that we care about. We want those to be interpretable, and kind of they are in terms of like they're things that we can talk about and people can sort of formulate sentences about, because in a way I just sort of started to do with my description of them. But we also need them to be identifiable. They can't like change wildly every time you rerun the model. There's got to be a unique solution to those. Notwithstanding the uncertainty you have in them, of course, but you can't find in a completely different part of parameter space a completely different rate of occurrence of a brain network state that equally well explains the data. You don't want that to be happening like in an uncontrollable way. So that's kind of the key issue, and the whole deep learning revolution is based on that not being important. So it's almost like that not being important is the reason why it's so useful. But of course, it not being important does depend on how you use it, depends on the application. So we have to think about using it carefully. So we only use it on parameters where the deep learning kind of approach, and we don't care about identifiability, but we just want the power of deep learning to find structure in the data.
Well, yeah, but
that's a very brief overview of my of how we're trying to work with it and how it dovetails with what I do.
But I guess what I meant is a lot of deep learning research nowadays is about interpreting those models, exactly because before people didn't care about it too much as long as it worked.
Yeah.
So I was wondering how those kind of methods that they use for interpreting the deep learning models themselves, how they can be adapted or applied to inform interpretability of brain-related things.
Yes. Well, maybe they maybe they can. We haven't taken that, but I haven't seen. I mean, I assume things like ablation and the stuff you've worked on, like permutation.
Yeah. Or for example, in a convolutional neural network, looking at the layers.
Yeah. Yeah. So I would say we aren't taking that approach.
Yeah.
Yet. I mean, we might come onto that and might find that it's useful. And other people do. So people, when they're doing computational neuroscience and try to understand how the visual system works, as you know, people will correlate the layers in the artificial neural network with the different areas of the visual system as you flow through the visual system. So V1, V2, V3 being related to different layers of a CNN, for example. So yeah, people do that kind of thing. We aren't doing that with the work that we do. We are the bits where we use deep networks, we leave them as black boxes, basically. We might explore, by generating data from that black box, some of the characteristics it has, but we don't try and go in and look at the actual parameters of the black box, whether it's a recurrent neural network or whatever it is.
Yeah, you have sort of other constraints.
Yeah. So maybe it's better like a concrete example of how we've used it in this technique that you know of, called Dynamo, which stands for dynamic network modes or modeling. I always forget which one it is. And that's like a replacement for the HMM using deep learning ideas. So the HMM has got this Markovian assumption. It's the final, the middle M of the HMM is hidden Markov model, Markovian, and that assumes that the state of the brain or the system that you're using the HMM on only depends on what's happening at the previous time point. And if you know that, then you've got probabilistically a full prediction of what's happening at the next time point.
Which is a strong assumption.
Very strong assumption to make, and as you might expect, it only captures short range dependencies. It kind of falls away exponentially in time in this case pretty quickly. So you aren't able to capture what we call long range temporal structure, and we know the brain's rich with long range temporal structure. It's doing stuff that you can sort of relate stuff that's happening at a certain point in time and stuff that's happening several hundred milliseconds or even seconds earlier. So that's a big deficiency of the hidden Markov model. So we wanted to have longer range temporal structure being modelled rather than this Markovian assumption. So we've used recurrent neural networks to do that, LSTMs, that kind of thing. And so in that case, the LSTM or recurrent neural network is capturing the spatiotemporal structure that allows us to predict the next state. But for the applications that we use it for, we don't need to know, or we haven't worried about the specific way in which that structure is being represented in the recurrent neural network. Yeah. And in fact, we know at least I think I know from the principles of deep learning that there's when you fit that model multiple times, the weights in that recurrent neural network will just be different every time. But the way in which it impacts on our state prediction should be the same every time. So that state prediction is identifiable and reproducible and interpretable as part of a sensible generative model. But then the weights and the RNN aren't. So having that combination of a module of our generative model being a black box, a recurrent neural network, interacting with other modules of our generative model, which are interpretable, like is a brain network state active or not at this point in time. That's kind of what we're doing. So it's that mix of non-identifiable, identifiable parameters in our generative model, and the deep learning comes in to help us do some funky stuff, but we don't try and identify what's happening inside that funky model at this stage. I mean, but we could try and do some things. You need to find summary measures from your recurrent neural network which were identifiable though for it to be useful. I think that's an ongoing battle for the field in general, I would argue, to do that. I mean, it works with CNNs because I guess the way in which the layers arrange themselves in certain applications are reproducible. But there's lots of other measures you could pull out which won't be. In fact, the vast majority, I would argue. It's then not identifiable.
So you are generally quite careful about using deep learning for specific parts which do not matter in the final interpretation of the result.
I'm sure this is my take on it. I mean, ironically, that's what has made deep learning so powerful, the fact that it doesn't worry about being identifiable. You just have shed loads of parameters, potentially more than the amount of data points, in fact, quite often, and you don't care where in that parameter space it falls, as long as the output from it is useful and reproducible, like classifies, a picture of a horse versus a picture of a dog. If it's doing that reliably every time, you don't care which particular non-unique solution it's found internally. It's just found one that's useful.
But still, it's interesting because researchers nowadays are able to tell how it's doing the classification. So, for example, how it's able to distinguish.
And that I guess there are summary measures of the networks where things are reproducible, and that's. I guess finding those is what those people do, and it's one of the challenges of working with these deep networks. And we haven't looked at that so much yet, or really very much at all. I know you've done a bit in your work by looking at that's more. I guess that's more the level of like ablating the input, so isn't it? So that's kind of like almost like before the black box.
So that's one way to go about.
So everything I'm talking about, I would still be happy to ablate inputs because the inputs are kind of identifiable, interpretable, whether it's data or it's part of the rest of my generative model. But yes, once you go into the network, then that's where the ablation. To work, I think you'd have to be quite clever at doing that in a way that was interpretable. You tried this in your work with some success, but not. I mean, when you try and look at the inside of the network, it gets a bit
messy.
Yeah.
I mean, that's definitely a challenge of using these deep learning methods. For your imaging data. Because I guess in other fields, you sort of like humans are able to tell what's going on. You see an image, that's normal. You see some text, that's normal. You hear audio. You can interpret all of these inputs, but in your imaging, what you see is time series signals which are not interpretable for humans. So I think that's a big challenge. In applying deep learning, because then you don't really know. It's like
you mean lack of ground truth, basically.
Lack of
or even subjective ground truth. That's true, and that's especially when we work a lot with generative models which are effectively unsupervised. Other than the data itself, you haven't got any labeling. In a lot of cases. Exactly. Although we do then apply it to
in tasks.
Or disease is the other kind of labeling we have. But things like the hidden Markov model is done at a level where we don't have labeling that level of temporal detail, and it's wrong to impose. If you want to tell what the brain's doing at very fast timescales, you don't want to impose labels from an experiment because they are too crude, and the brain is too variable temporally to synchronize with those cues from the external world precisely. And so that's why we still use things like the HMM and this new technique, Dynamo, even when we have got task timing information, because the brain doesn't align itself perfectly with that timing. It will vary from trial to trial. And so you want a technique that can adapt to that, which is therefore going to have to be unsupervised. And then afterwards you can sort of see how that relates to the timing of the task. But you'd lose detail if you did that from the word go by using a straightforward supervised learning approach.
Yeah, there's a lot of variation, there's a lot of noise.
Which is why we go for these unsupervised generative model-based approaches, and then post hoc, downstream, link them to the labels that we have in time.
Mm-hmm. We've been quite technical so far, so I'm going to switch a bit to a different
That's what I'm most comfortable
topic.
talking about.
Okay, so that's... I was curious, how do you navigate working across so many different projects and all the context switching that you do? Because to me, you always seem up to date with everything.
That's very kind.
Well,
yeah, I mean, it's difficult. I mean, you have to. I mean, the most important thing that I do is partition my time. I'm not very good at multitasking. If I try and do 3 things at the same time, it doesn't work. So you have to basically just, devote yourself to something for a good slice of time and make sure you do that well before you then switch away from it to something else. For me, for example, I'll switch off email for like a few hours in the afternoon and not even look at it at all, so that I can concentrate on reading a paper or whatever it might be. So that's probably the first thing that pops into my head in answer to your question.
So you're a sequential process.
Yeah. I don't really, certainly with stuff as in sort of, for me, intellectually involved as this, I just couldn't do it any other way. Mm-hmm. I get distracted a bit too easily I know I might have an email coming in, or God forbid you've got notifications popping up on your computer screen. And then other than that, it's just accepting that you have to do certain things in a job that are less attractive to do. Certainly when you get into more senior roles. And actually as you get into them, you find them perhaps more enjoyable than you thought they might be anyway, just because they're kind of interesting challenges.
But I guess what I meant is specifically the research projects you're working on. How can you stay on top of all of them and switch between them and
I keep notes.
Okay.
I have online notes basically, and if something like if a penny has dropped in a supervision meeting, after the meeting I'll put it in my notes. I don't tend to do it during the meeting; it's more after the meeting. Or if I've been confused by something in the meeting, either that it's explicit or something I've kind of kept quiet about because I don't want to embarrass myself, then I'll doesn't happen very often. Maybe it does. I'll then go away and think about it and then make some notes again. And finding time for that is quite challenging, obviously, when you've got a busy schedule. But so my notes are quite important. And it's not even that I look at the notes that much, just the act of making notes is useful. It makes you sort of self-check that you have kind of understood what's going on. And if you don't, and occasionally they are useful to look at though, like, if I've not seen somebody for a week, which is quite common with supervision, and I've not thought about it in that week, it is worth just to, there's like a concept which I can remember I worked out, but I can't remember the details. I go look at my notes. It's like, oh yeah, crikey. And without that, I'd have to be almost back to square one again. And so you're much more cumbersome with keeping up to date, as you said.
Yeah.
So the notes are probably the main thing that I use. And it's when I feel like I need to, spending some time after supervision or between meetings, just having a bit of a think about it. And there's certain problems that I'll get deep into. Like there's a new model I want to try and work with, implement, and TensorFlow, whatever it might be. Then I will get hands on with it in a toy problem setting. It just it makes a huge difference doing that. And then there's areas which I'm become more familiar with where I don't need to do that because I sort of know how it mostly works. So I don't have to be doing that. But with new stuff that's new areas, then I'll have to do a bit of that as well, which is frankly really enjoyable. So I'm quite happy to do it.
So you enjoyed the context switching?
No.
No. Okay.
I'm happy when I'm just ploughing away at something for like a whole day. It's like really, getting deep into something.
Do you think you can do the same level of deep work that I guess you've done, for example, when you were doing research alone, or is it less, is it more?
I've made a real effort to try and, I went through a period a couple of years ago, sort of around COVID, maybe just before, I was really struggling to do that. Maybe just through probably a lack of time management and purpose. But so I made a conscious effort to try and free up time to do that sort of thing, have that time to properly focus on something and get hands dirty with writing code even. And that's much happier doing that. I'm a better supervisor. I can help much better. I can stay up to date with what's happening in the field much better by actually being, rather than just reading it and it's all theoretical. I can actually play with it, and so that's been quite an important change. And it helps me enjoy my job. It helps me do it better as well. And I do have periods in the week where I keep free to do that. Mostly Fridays. That's a secret. Don't tell anybody.
So no meetings on Fridays?
I try and avoid meetings. Unless there's a meeting that has to be on a Friday, I will say no. I basically effectively I'm busy on a Friday. Yeah. Because and I am really. It's important for my job. Okay. It's important for my sanity.
So you're not just taking Friday off?
No. My wife's always cursing me for still working at like 6:30 on a Friday evening when the kids are kind of requiring some attention and whatnot. Yeah.
Strong question coming up. What do you think was the biggest failure in your academic journey?
Probably the amount of time that I spent working on group stats in fMRI. Quite quick to answer that, wasn't I? Didn't hang around.
How was that a failure?
Well, it wasn't a failure. It resulted in something that essentially worked, but it just wasn't worth the hassle. It wasn't worth the effort. So this is like post PhD, early postdoc working on fMRI group stats, and it was just like there's some deficiencies with the way it's done classically in those days, and it was a Bayesian approach, so that was great, and that's what lured me into it. But we were trying to marry the sort of Bayesian approach with classic frequentist statistics in a way that went to a level of p-value accuracy that was just not worth the hassle. Mm-hmm. Yeah, and that when you start chasing p-values, that's when you're in trouble. Like trying to, and the people that work on that and enjoy it, fair play to them. I personally don't, and this is where I was working in that kind of area and spent far too much time trying to get this technique to work in a way that was, unbiased statistically, gave the right number of false positives, the correct p-values. We had a good sensitivity, mm-hmm. And was computationally efficient, etc., and it just wasn't worth it in the end.
I mean, in hindsight, it's always easy.
Yeah, and it's one of those things where you just kept sticking at it because we've almost done it, we've almost got it, it's almost there. So I ended up working on it way too long, and it becoming not fun to work on. And it's in FSL, this software library that we have for fMRI, and has been used, I think, quite a lot. But it just wasn't worth the effort.
Yeah.
Is that the kind of answer?
I mean, if that's how you feel, that's the answer, yes. So on the flip side of that, how do you think one can achieve greatness in the pursuit of science? As maybe an example. Well, anyway.
Don't work on frequentist statistics, unless you love it. Yeah, I don't know. I don't have any. I don't think I'm a scientific great, first of all, by any stretch, or I wouldn't describe myself as anywhere near that kind of domain. I mean, it's just about finding something you're interested in and you enjoy. I mean, that's for me. I focus more on what finding a line of work, an area of science that you enjoy working on, and that someone's happy to pay you to work on. And if you've got that, it kind of doesn't matter how great you are or how sort of well known you are or what the impact is of your work. Obviously, it can add some satisfaction, but if you're enjoying it without the impact, then that's fine as well.
Yeah, I guess. I mean greatness not in the sense of impact, but more in the sense of true general discoveries, or like the classical things we think about when. Yeah.
Okay. I mean, you can pick the right question to work on. I mean, I've had the other people say the most important thing in science is working out the right question to work on. I'm not so worried about that, because I think it's quite hard to know what the right question is. Even like, obviously with hindsight, you can sort of give the impression that you knew. I think it's better focusing on things that you find interesting to work on. And those two things might look like the same thing, but it's easier to focus on something you enjoy working on and are interested in working on. That's a tangible thing to try and achieve. But I think it's hard to sort of know if that's going to be a question that's going to lead to some big breakthrough that's going to make a real difference. You can have a guess, and it's quite likely that sometimes the interesting questions would also have a chance of doing that. But yeah, I'd rather focus on what's interesting and what's I'm inspired by to work on.
Do you have a sense of what role intuition and creativity play?
Yeah, quite a lot.
Progress?
Yeah, just blind luck. Yeah. Yeah. I mean, for sure.
From your own experience.
Yeah, I mean, it's not just. There's luck, obviously, where you land and what you start out working on, and then you take a punt, taking up a certain position, and a lot matters on that. I think a lot rests on having good people around you as well. Like if you work with interesting people who help to inspire you and bounce ideas. Yeah. It gets harder as you get more senior, because you spend less time interacting with people who are your peers and more time interacting with people in your group. But yeah, luckily you get some great if you're lucky, you get some great postdocs who fill that gap, and PhD students. Sorry.
Thank you.
She said that. Or earnestly meant as well.
Yeah.
So that's. But yeah, in terms of like intuition and what's the other word you used? Creativity. I think they're. I mean, intuition's a bit of a funny word, isn't it? I don't sort of know often how to think about that word.
It's not very well defined.
It's a bit like free will.
Yes.
Like ill-defined and therefore a bit pointless to talk about. I guess a working definition of intuition for me would be like, you're not just having a random punt. It's based on something that you've got some structure to your understanding of a domain, and you're going to use that your understanding of that domain to then extrapolate beyond what you know. And the way you extrapolate is kind of your intuition, I suppose. It's like, so it's not uninformed. It's taking a calculated guess, I suppose, is the other way of describing it. So that's an important thing. I mean, that's the only way you come up with new ideas, really, apart from just being completely random. You base it on what you know. You might base it on what other people know by reading around and chatting to people, which then becomes stuff you know. And then you sort of think. A lot of it is putting ideas together. I guess that's where I've often worked in that way. It's like, there's this idea over here that might be outside neuroscience completely, might not be. And this is idea in neuroscience or neuroimaging that we're trying to do, put them together. Oh, crikey, that was quite interesting. So that mixing of different concepts is often how kind of my replacement for intuition, if you like. And that's less of a random punt. If there's benefits you can see here in this one idea, and benefits you can see in this other idea, and those benefits look like they're going to be complementary, then you plug them together and see if it works.
Yeah, it's good to have, I guess, your background as well, engineering, computer science. Because you can identify these kinds of ideas between domains.
That's true. That is very true. But it's quite common in neuroscience. It's quite a multidisciplinary field. People come in with their different perspectives, whether it's a medic or a biochemist or whatever it is.
Would you give that as advice to young researchers, for example?
What, the plugging together of random things?
Yeah, or
to something new.
To have experience in multiple fields rather than just sticking to the same thing.
I don't think it's necessary, but because you can learn on the job. If you've got the passion and you're given the freedom and the time to do it, you can learn on the job, which is what PhDs are for example, and some postdocs. So, so you don't think you need to do that, but it can help. Yeah, one variant on that question is it important to go to different labs and experience different things? Clearly, I've failed miserably to do that, apart from moving between fMRI and OBAR, between fMRI and MEG and EEG, and still doing fMRI as well. But yeah, that kind of, I haven't been outside Oxford in my career.
Well, you went to IBM.
I went to IBM for a couple of years. Yeah, that is true. And so that's another example where people say, oh, it's really important to experience a mixture of environments and labs. But again, it's not. I'm sure it does help a lot of people, but it's not crucial. And that's how I'd answer your question as well. I think it can be useful, but it's not vital. It's not a necessity. So I wouldn't say to someone, you must mix up your disciplines. You must work in different labs. I would rather they just try to make the next best. It's better to make the next best call just based on merit in terms of like, am I going to be working on something fun, interesting? Am I going to be in an inspiring environment? Does it lead on to is there potential beyond it? Am I going to be going down this dead end? Those sort of things, and that's kind of like orthogonal to whether or not it's in a new lab or in a new discipline. And those override the new lab, new discipline questions for me.
So I guess your core advice is do something that you're really interested in. Yeah. Always. Yeah, which put that as your primary.
Yeah, thing. If you can. I mean, obviously, pragmatism kicks in sometimes. You need to get a job sometimes, or you're happy to compromise on certain things. So maybe, but otherwise,
a bit more philosophical about AI and the brain. Do you think we can achieve a level of AI or this kind of machine learning models that can accurately model and simulate human brain activity? I guess we touched on this.
Do you mean like artificial human level intelligence, or do you mean just like really sophisticated deep learning for understanding brain recordings?
I guess the brain activity itself, to be able to model it fully, whatever that means. Or is that this computational irreducibility that you mentioned earlier?
Well, so obviously with more data, and we can then work with more complex models and get more detail. So that I think that will just carry on happening. The one caveat to that, which I'm sure you've heard me bang on about before, is dealing with the between subject variability when you do that. So that's an important part of it. But again, these big models can potentially do that for you in principle.
Yeah, because I guess when I say human brain activity, there's a question which human brain.
Yeah, we know there's massive variability. So with these bigger models, they in principle could deal with that variability either explicitly or implicitly. I think there's room for working to ever more detailed levels of description of the type that I've worked on, and potentially that might then provide enough detail to do something more meaningfully with these more biophysical models, where you run out of, where you kind of hit the limit. I mean, the limit is when you end up having with brain. If the brain imaging devices don't improve, you'll hit a limit of what's the signal content in those recordings. Like we only have a certain number of sensors around the brain, and actually you can do simulations to show that adding more sensors wouldn't necessarily help because it's still going to be outside the brain. You hit that limit at some point. Even if you've got, like, let's say, hypothetically infinite amounts of data on infinite amounts of subjects, you'll hit a limit to the information you have access to. But I think that's a long way off. I don't see that limit being hit anytime soon.
At the moment, you feel like the limitation is more on the software side, on the data side, like on the amount of data side?
Yeah, I think the amount of data and the ability to compute it. But that's obviously the deep learning is helping with that. With MEG, we haven't got enough data. We need bigger data sets. fMRI, as you know, has got some really big data sets, which we'd love to have in MEG and even EEG.
Yeah.
The EEG, there are big data sets, but they're not of the right quality. They're not of good quality, so that's a big problem. Whereas the fMRI, they've got various studies, but the obvious one is UK Biobank, where they've got pushing up towards 100,000 subjects with fMRI scans and a bunch of other MRI and a bunch of other non-imaging data. So it's an amazing resource, and we have nothing even close to that with MEG or EEG, which is a real shame. Well,
it's a waste of money if you ask.
That's a real shame, but it kind of represents the dominance that MRI's had in the field of brain imaging for the last 20 years. And there's a reason for that. So I'm not dismissing it. I'm just saying that it's a shame there isn't a bit more breadth of interest in other ways of imaging the brain at that level.
So we're still quite far off of hitting the hardware limit. and it's more on the data amount that we're currently
and quality, of data. I think that we'd obviously need to create the computational methods to go along with that and to work with that data. But yeah.
Do you have a sense of when we hit the hardware limit?
No.
How can we progress from
I'm sorry, how can we bet? Well, you need new modalities, don't you? That's it. You need to. I mean, my hunch is that this limit is sufficiently far off that we will have come out with new imaging modalities by then. But who knows? At the very least, you might have to start working with invasive measures which you can get in patients, for example. So, there are variants of MEG that you know about, like optically pumped magnetometers, which may provide some improvements, and are likely to provide some improvements in the next few years. But they're not the kind of paradigm shift that perhaps you're referring to.
Yeah. I guess this ties into neurotechnology more generally. And what is the future of?
I mean, we do. It would be great if there were a new way of recording brain activity without doing surgery. It's hard to see where that would come from, but then that's often true in science with technology.
What do you think is more likely, though? Like some kind of progress in surgical methods where it's going to be so easy to do and seamless that invasive is...
that's the Neuralink kind of perspective, isn't it? And other companies as well,
So is that more likely to happen, or a completely new kind of non-invasive modality, which we can't even think about right now?
I mean, who knows? I mean, I think that it's probably more like... Well, both things are possible. The one that's perhaps feels like it's more likely that it could happen is the invasive one, just because people are already working on that. So it just feels like it's more achievable. You sort of know what it might look like. And it would look like people using it in disease applications. And this is not me saying this, is just parroting what people like Elon Musk and other people have said who work in this field. You first of all apply it to clinical disorders, but then in the process of doing that, those people then might see other benefits of having the technology. So you can develop that sort of non-disease aspect of the technology, and then that just either the big jump then is that explodes and it's like, wow, look at this is amazing. Suddenly I can do X and Y. Then people are motivated to have surgery done on themselves. So the motivation's there. Then also hopefully you make the procedure sufficiently safe that the motivation overrides any remaining risks of doing the surgery, and then it could take off. But I mean, it's a bit sci-fi, isn't it? The idea of the majority of us, or
but it's at least
non-disease people having brain surgery.
At least it seems like there's steps leading.
Exactly. There's a roadmap you can see for it.
Whereas for the non-invasive side,
I can't see the roadmap for that.
You need some kind of core physics. Development. I'm not even sure.
It's like you need some new application of quantum mechanics or something, or some new area of physics to be discovered that would make it possible.
For example, FNIRS has been quite interesting to see, because just the fact that you can shine light into the brain and that reflects off the blood vessels, and it provides you a measure similar to fMRI.
But nowhere near as spatially accurate.
Well, I think getting there.
Okay, I'm not up to date with the very latest. Last time I looked into it a few years ago, probably 3 or 4 years ago, it was still looking pretty crude. And certainly can't go deep brain structures like fMRI can.
It's more cortical.
Superficial, cortically superficial,
So what do you think about, I guess, what you mentioned, Neuralink and this kind of vision of merging in the far future computers and and brains?
Well, I've kind of already...
what are the implications of this?
I guess I've already talked a bit about it. I mean, who knows? It's not something I work extensively or at all on, really. So it's more based on my idle thoughts or hearing the odd podcast, people talking about it themselves. Yes, the roadmap is as I described, how you might get to a point where non-disease people might have it done. What's possible if that when that happens, or if that happens? Yeah, I think that's one of those things we just don't really know. And I guess a lot of it would depend on the bandwidth you can achieve between the devices and the brain, the cortex.
So the data transfer.
Yeah, I don't know enough about that. I've heard some people say that the bandwidth's really poor, and then you hear other people saying the bandwidth's going to be massive paradigm shift compared to what we do at the moment, which is vision and speech, and certainly in terms of our output. So at the moment, our output is it's like mouse and keyboard or speech, isn't it?
Oh, yeah, it's limited. It's
very limited bandwidth, and I've heard some people saying that will be the big paradigm shift. That suddenly our output,
like the information transfer rates.
Yeah, exactly, will be massive. But I've heard other people say that it's the sort of technologies that are being looked at is still really poor. But so I haven't looked into to know what the truth is. So I'm assuming, let's assume the bandwidth's big, then yeah, I mean you would envisage people being able to interact with computers in a completely new way. And then obviously there's the closed loop aspect as well, where it's going to be one of those complex and adaptive system type situations where it's almost going to be impossible to predict what's possible and what's achievable. I guess some bad as well. So that's some bad stuff as well. That's
there's always bad.
Yeah, it's not just the surgery that's a risk, it's also the impact it might have on your brain activity. But I think people will be prepared to take that risk. I think the surgery is more of the barrier than the impact it might have on your mental health or your neurological health. So yeah, who knows? I mean, the obvious thing is like massively improved efficiency with interacting with computers and communicating with computers and with each other via computers. So that's the obvious thing. Obviously it might have impact on our mental health and on both, hopefully good ultimately, and also in disease as well in a much richer way if this kind of higher level interfacing of computers and brains is achieved. And I suspect if that does happen, it'll be again in a very black boxy way. It'll be done via some objective function of I want this to be done, some identifiable output that's quite high level, and then everything has to be a black box.
Using a black box.
Yeah, so you actually probably need that much in the way of a neuroscientist to help you with design or stimulate at 250 hertz in this particular area of the brain. I mean, I think that's one of those things that people are trying to do at the moment, but I think longer term will just become a silly way of trying to approach the problem.
Yeah, speaking of closed loop, how much have you worked with stimulating? So the other way, going the other way, because I feel like that's more, it's in infancy compared to recording from the recording activity.
Yeah, well, again, it's a bit related to the lack of invasive. I mean, apart from in certain very specific disease populations, you haven't got the invasive capabilities to really do good old stimulation in very specific ways.
Well, there's also non-invasive stimulation.
So you're stuck with non-invasive instead, and until recently, they're very spatially crude. The ultrasound stuff that's coming on board the last couple of years or more is looking like it's more spatially focused. So yeah, I don't know enough about that to sort of be able to extrapolate where that might end up, but that's got some potential. So yeah, I mean, I don't know enough about the that technology, and the other technologies are a bit spatially crude. So that's kind of held back what they're able to achieve. So things like TMS, and
transcranial stimulation, and
TDCS and TACS, which are electrical currents rather than magnetic stimulation. So, there's a decent amount of research being done. TMS is being looked at for helping with depression, for example, but it's spatially crude. So the only thing you can really play with is a temporal signal, and there's more to do there. I think, like to actually have a closed loop where you stimulate in a particular way temporally at a particular point in time, albeit spatially crudely. I think there's an untapped potential there, because people's understanding of the dynamics have been quite poor until recently. So yeah, that's and then with the black boxy closed loop system, you might be able to leverage that, and that hasn't been fully explored, I don't think, at the moment.
To adapt the stimulation.
Yeah, temporally in particular. Yeah.
Some more closing philosophical questions. What is something you believe that most people would disagree with?
Something I believe that most people would disagree with. Crikey. That's a hard one. I mean, I can get political if you want.
Yeah.
I would ban all private schools. Is that the right
Is that something most people disagree with?
Well, I mean, it would have happened if most people agree with it, wouldn't it? So, that's, I mean, yeah.
So why would you ban all private schools?
I believe in equal opportunity. I heard someone talking about mental health being should be like a fundamental human right. You know, I mean, your mental health being considered, or not being neglected. That should be a fundamental. I think equal opportunity should be a sort of fundamental human right in a kind of similar way. And there's no excuse for not trying. I mean, you can't achieve equal opportunity, obviously, but it's an ideal you should strive for. And we don't even bother trying, certainly in this country. And I think a lot of Western countries, America, for example, people don't even try and strive for equal opportunity. They almost actively keep things going that do the opposite. And I think that's such a. It's not anywhere near as bad as some of the historical nonsense we've been up to as a species, but. I guess because I've got kids, it's one of the things that grates.
You notice more.
It grates with me a bit,
Mm-hmm.
And also when you see, just the division between rich and poor in this country, and worse in other countries, and that's just maintained via this, this total head in the sand approach towards equal opportunity. I don't want equality. I want equality of opportunity. Just to be really clear. Yes. And private schools get in the way of that. That's probably the easiest fix you could do. Why, just because your parents have got money, should you be given more of an opportunity than anybody else?
Mm-hmm.
You shouldn't be actively encouraged. You can't stop that happening to some extent, because money is going to be an inevitable part of our society. But you can try and do things to sort of mitigate for that impact of people having more money than others, and by taxing the hell out of private schools, or ideally banning them, you'd do a lot to help with that. And also you'd find overnight you'd get a massive improvement in non-private school education as well. So everybody else has to put up with this bargain basement version. It would be a lot better.
I mean, to me, the education system just seems so inefficient in general, especially with, it's a bit cliche to say, but with AI progressing so much lately, and the things that you can do with AI now, like integrating into education and leveraging it. I mean, it feels like even the internet has barely been integrated into the education system and computers, but now so much progress is happening in AI, and nobody's gonna provide, kids a more efficient way of.
Well, again, if you did have competition, and it'll be happening more in private schools. They are making use of computers, and it's happening in state schools. To be fair, I mean, my kids have laptops that they use, and the internet course, and online courses and online resources. So it does happen. It's just it's just obviously nowhere near catching up with what's happened in the last few years in terms of what's possible with AI, and they're having to scramble to deal with things like ChatGPT. So it's definitely going to be a period of fast change, but the private schools will adapt more quickly and more effectively. You saw this in COVID, in lockdown. Everyone's like working out what to do with their kids and what level of support they were getting at home during lockdown, and the private schools were just all over it straight away. I mean, generalising, of course, there's going to be exceptions, but generally speaking, the private school kids were getting things sorted out pretty quickly, online lessons, resources, whereas the state school wasn't really that well prepared for it. Didn't have the computer systems in place, didn't have the kind of resources to make it happen quickly. So it's an example of how they'll be slower to react to things like the current changes in AI.
But then if you ban private schools, how is that going to make it better?
Well, because the money that's currently being pumped into those can be pumped into the general state system instead. Or what might then become the private school for everybody state system. So schools are run by private companies, and you get given like universities. Yeah, you know how universities are run in this country. Surprise, surprise, our universities are quite successful.
Yeah,
you basically have capped fees. And when I was a student, I actually was given a grant for I chose. Well, not chose. Obviously, you had to get into the place as well. You had to get selected, but of the places you're selected to go to, you then choose which one you go to, where you're going to take your pot of money that you've been given by the government. Why not do that with schools? Get competition, the money can flow to everybody into the best institutions that then have to try and compete and keep up with each other.
Yeah, that seems to work quite well at the PhD level, specifically.
Well, and the undergrad level as well. I mean, it's not perfect again. And the main reasons why it's not perfect are actually the knock-on effects of the lack of equal opportunity at the school level. So, like, for example, the bias of more private school,
so it should be started
private school
earlier.
Kids going to Oxford or Cambridge, Oxford and Cambridge, and the other top universities get all the flack for that. But it's like they're not the problem. The problem is the schools.
Yeah.
The problem's happened before they turn up at university.
Yeah. So the equal opportunity is applied too late, right?
It's laughable. I can't believe. Whenever I read an article about the university's vice chancellor having to defend the current ratio of private school to state school, it's like, I mean, you just totally focus on the wrong problem. It's the schools you've got to focus on, the lack of opportunity and the unfairness of the private school system. Sort that out. And I'm back on that again.
All right, closing question for you. What would you say is your life philosophy?
My life philosophy? It's meant to be a concise answer.
No.
What's yours?
What's mine?
Yeah, help me out. What's yours?
Well, I guess what I mean is like principles by which govern the way you live and goals and reasons why you do certain things.
Well, the reason why I'm struggling is quite a multifaceted thing, isn't it? Like it's like life's a bit like an optimization problem, like a hyperparameter optimization problem. Like you try and obviously it's always changing as well. But you're trying to for me, get the right balance of a whole bunch of different things, both in terms of what you interact with in life, who you interact with, what you do for your job, what you do for your family life, what you do socially, what you do physically, exercise, what you do health-wise. Just everything. It's trying to get everything balanced in a way that keeps you sustained and happy and fed. I guess I know what I should say. I'm always banging on about this to my kids. The most important thing in life is, I think, balance. For that reason, you should never go to an extreme on anything, whether it's a diet or it's some aspect of how you exercise, even, or how much time you spend doing something. You want to have diversity of things in your life. Don't take something too seriously. Don't take it too trivially. Have a mix of different types of friends. Spend good amount of time at work, but not too much work. Too much time. Spend time with your friends, but don't just only do that. Just I think having a good mix of diverse things that hopefully are generally good for you, but in balance. Because having too much of one thing that's on the surface good for you ends up being bad, I would say. And even to the point of having, you don't want life just to be a hedonistic sort of, whatever, I'm just here to enjoy myself. Because that's a lack of balance as well. Life needs to be a mix of frivolous enjoyment, but also challenges and facing some hard stuff, because that gives more meaning to the frivolous sort of hedonistic stuff, makes you enjoy it more. And then that stuff gives you the fuel and the space to then be able to handle the difficult stuff. So I think balance, mix of stuff.
So sometimes you do want to focus on certain things more, and then Well, thank you very much for having this
Thank you.
amazing discussion.
Yeah, it's been very interesting, taking me into areas I wasn't expecting. But yeah, thanks very much.